So today we're going to take a sample of that mystery bacillus and we're going to go through the gram staining procedure. So here's that colony that we're going to sample. I've got my needle, I've got my sanitizer, got all my stains ready. Here we go. So the first thing I'm going to do is I'm going to take a small sample with the needle, kind of just scoop it out, and we're going to make a smear plate here. We want to smear the bacteria over the slide so that we can expose a lot of them to the stain. Since this is a dryish sample, I'm adding 1 drop of water, and we're going to swirl that around to spread the bacteria out. And I should have done this first, but we're going to draw a circle around this so we don't lose it. Once it's dry, we come back, and we're now going to heat fix these bacteria by passing them over a candle flame. Heat fixing helps the bacteria stay on the slide as well as kills them so that they're ready to stain and they're not going to go anywhere when they go through all these washes. Our first stain's crystal violet. We're going to put some on here into the circle. We let it sit for a minute, then wash away. And that's just water. Second is iodine. Same thing. We're going to put a couple drops into our sample. You can see some of the sample remains purple. And we wash that away after sitting for a minute again. This decolorizer step will decolorize anything that is not purple, not holding on to that stain at this point. And we also wash it away, but I didn't show it. And our last stain is safranin, which is the red counterstain. And we're going to put drops on there and let them sit for another minute. Finally, everything's washed away. So we also want to blot this dry. We don't want to wipe it because we don't want to wipe away our sample. Gram staining ends up with either gram-positive or gram-negative, and gram-positive bacteria are going to be purple with gram-negative being red or pink. We were expecting to see a lot of purple, which we kind of do, because bacillus species typically are gram-positive, but we also see what looks like little endospores, which can be survival structures for bacteria. Those guys don't stain in the gram stain because they have a more impermeable outer layer.